Welcome. I hope that all of you have utterly enjoyed the metabolism coverage that we have already completed and that you're super jazzed to dive into gene expression. Our goal right now is to begin to think about genes, to define the genome of a cell, and then to look closely at the central dogma of gene transfer. Let's do that by checking out Norbert. Now, Norbert is experiencing social isolation at the lab, and I, of course, am doing it at home. So Norbert is not physically here, but I did want to draw a brief picture of Norbert so you could zone in and look at the genome and recognize that the genome is a combination of the chromosome and the plasmids. Now, just to make our view a little bit more accessible, we're going to simplify by creating a zoom-in version and actually flattening the chromosome out on the page. So we remember that in doing that, we remember that the chromosome is a single circular piece of double-stranded DNA. So I'm showing both strands, showing that it's circular, and recognizing that as the chromosome. Now, technically, the genome is just defined as a combination of the chromosome and the plasmids. So now we can take a look at the plasmids, and we could also zoom into those. So if we just said chromosome plus plasmids, let's just do that. We know the plasmids are also singular circular pieces of DNA. So if we're zooming into those, we know they're much smaller than the chromosome. And there may be more than one of them. And we know they're not required for replication. They're extra chromosomal, but they often carry superhero endowing powers, right? Now, one thing that's very important to note is that despite the fact that even many textbooks inaccurately describe the genome, it is really important to note that it is both the plasmids and the chromosome. Many times even textbooks will interchangeably refer to the chromosome and the genome, calling the chromosome the genome. But technically that's not the case, that there are genes on the plasmids that do contribute to the overall genotype of the cell. So we know that on the chromosome and on the plasmids, we've talked quite a lot on the plasmids about genes that might be found there. For example, the gene called the beta that encodes for the beta-lactamase enzyme, the enzyme that cleaves beta-lactam antibiotics like, for example, penicillin. We know that's one particular gene that can be found on a plasmid. So the funny thing is that we all know about genes, right? We talk about them all the time. Probably within the last week, you've said to yourself or to someone else that something is genetic. But what's really interesting is to know that we don't actually have a great definition for a gene. And in fact, depending upon the source that you look at, gene will be defined in different ways. So sometimes people take sort of the cop-out definition, which is that a gene is the smallest functional unit of a chromosome or a plasmid. You could also define a gene, and for our purposes, we're going to define it here as being something that encodes for a product. Now, that product may often be a protein. We talked just a minute ago about a product that could be an enzyme, that the beta-lactamase enzyme is a protein, right? So encodes for a product, often a protein. However, it's not always a protein. And that's where I challenge you to think about what is a product of a gene that isn't a protein. So one really great example is a tRNA. So a tRNA does its job as an RNA. It's never converted to protein product. Its whole job of being an adapter, matching amino acids to their corresponding codons, it does that job as an RNA. There's also a ribosomal RNA or rRNA. Very much a product, that's a little r, very much a product of a gene, but certainly never converted into a protein. So we recognize these as things that could be made or encoded for by a gene. Now, even RNAs can have enzymatic functions. They're actually called ribozymes. Check out the work of Thomas Cech in the 1980s at CU Boulder. Really amazing, right? RNAs can be catalytic, but they're never converted into proteins. So hopefully this helps you to get your bearings when it comes to recognizing what the genome is and what a gene really is. And now I want to take your time to review the central dogma of gene transfer. So this idea of a gene getting converted into a product requires processing. So genes can very much, and so what I think we'll do here is let's pick a spot on this chromosome and let's make this dark purple gene right here. And then let's actually make kind of a blow up picture of just this region there of the DNA. And we'll just draw it in simplistic form. So remember 5 prime, 3 prime, coding strand of DNA, 3 prime to 5 prime, template strand of DNA. And then we have our lovely purple gene. And that gene can get read by an enzyme called RNA polymerase, the builder of RNA. And then that can get used to make the RNA. So the RNA is built in a process called transcription. We know that RNA could be mRNA, it could be rRNA, it could be tRNA. If it is mRNA, we'll just say it is for good measure. I'm going to erase some of this so we have some room. Then that mRNA can get recognized and read by a ribosome and converted into polypeptide chain. So that can be used, that mRNA can be used to make protein. And you might remember that that is a process called translation. Now, one thing is missing from all of this, and that is that DNA itself has to be able to it needs to be able to copy itself, right? A plasmid and a chromosome, whenever a cell replicates, when it goes through binary fission, it has to be able to copy itself, and that's a process called replication. So we're going to dive now into looking at the very dirty details of the process of replication. After we do that, we're going to dive into transcription and then finally into translation. Together, this is all called the Central Dogma of Gene Transfer. Now, this sounds simple when you look at it this way, and you might be thinking, man, Rachel, I learned that in kindergarten. But please recognize that as we dive down into the details, we're going to start to get to the nitty-gritty, and it's going to be harder to recognize what little things play a role in these separate processes. So pay attention and keep bringing yourself back to the really big picture as we get down into the nitty-gritty.